Outcomes in relapsed ALL are extraordinarily poor. Whether we talk about first relapse or, as was enrolled on this trial, patients who are a second relapse or beyond, or who are primary refractory, we're talking about a life expectancy of around 5 to 10%. This is based on the MRC ECOG data. This is also based on institutional data from MD Anderson. Even when we achieve CR, even when we take patients to allogeneic stem cell transplant, the best we do is if we can catch them at first relapse and take them to an allo. Short of that, we save almost none of our patients. Things are getting better in the era of inotuzumab and blinatumomab, but I still think, particularly for those patients who are now, as again, enrolled on this trial, who are beyond first salvage, we're struggling. There's no question here that all patients who fit into this relapse refractory category for acute lymphoblastic leukemia are a major unmet need. KTE-X19 is very similar to the product that was approved for diffuse large B-cell lymphoma. It uses the same CD19 murine scFv coupled to a CD28 co-stimulatory domain. Where it's distinct from that product is the KTE-X19 depends on T-cell isolation. So we sort out any circulating lymphoblasts that were collected at the time of apheresis from the manufacturing. So we then proceed with sorted T-cells for the viral transduction and also for the T-cell expansion prior to infusion. To enroll on the ZUMA-3 trial, patients had to be either primary refractory or relapsed after 2 or more lines of therapy. We did require patients have morphologic evidence of disease. In other words, all patients had to have greater than 5% blast in order to enroll. We did not enroll anyone who was MRD only or patients who had only extramedullary disease without bone marrow disease. Short of that, the inclusion criteria were fairly open. Had to have a good performance status, so ECOG of 0 or 1. Had to have good organ function, meaning we couldn't see grade 3 or grade 4 hepatotoxicity, renal toxicity at the time of enrollment. Patients with prior allogeneic stem cell transplant were allowed to enroll, but we simply said that they had to be greater than 100 days from the transplant with no severe GVH. Similarly, you could have bone marrow involvement with extramedullary disease. However, we simply said that patients with high burden or symptomatic CNS disease should not come on. So this is actually the end of phase 1. So what we're seeing at the end of phase 1, we had 3 dose levels that we explored. 2 times 10 to the 6, 1 times 10 to the 6, and 0.5 times 10 to the 6 cells per kilogram. At the target dose that we're taking into phase 2, the 1 times 10 to the 6 cell dose, we demonstrated an 84% CR/CRI rate. In terms of durability, we will show at this meeting that 12 out of the 16 who achieved a CR or CRI remain in ongoing remission. We did attempt to look at the role of stem cell transplantation. In total, 3 of these patients did undergo stem cell transplantation following CAR T-cell therapy without a clear impact on the duration of response. I'm very excited to share that the modified toxicity guidance that we employed and will be sharing again at this meeting significantly curtailed the neurotoxicity rates. What we're looking at now is a grade 4 neurotoxicity rate of 0 and a grade 3 neurotoxicity rate of 11%, which was 1 out of the 9 patients that we studied at the 1 times 10 to the 6 cell dose with this modified toxicity guidance. Specifically, what we did is we restricted tocilizumab to the management of CRS, not neurotoxicity, and strongly recommended the earlier institution of steroids, that is at grade 2 neurotoxicity rather than waiting for more severe neurotoxicity to emerge.